E&M and is the editor-in-chief of JBMR. He has research funding throughout his long career. Currently has at least 4 NIH awards where he's PI or co-PI, and also a Department of Defense grant. He has extensive knowledge and work that he has done in the field of bone biology. And today he will be talking to us about N-cadherin in tumor-associated osteolytic plus cells, restrains tumor growth via modulation of TGF-β Paracrine loop. Dr. Civitelli.
Well, thank you very much for the nice introduction and very nice words. And welcome everybody to this seminar. I hope I can give you an idea of our work, current work on the relationship between bone cells and cancer cells in the tumor microenvironment. I know this is a little bit away from general endocrinology, but it's the problems related to how to manage cancer patients, especially breast cancer and prostate cancer patients, is becoming more of an issue related to endocrinology because of course, the endocrine therapies these patients have to take for a long time. So let's see if I can, okay, move on. So these are my disclosures. And these are my credits, just to make sure that I put them up before I forget at the end. I primarily thank the current members of my lab. Toshi Sugatani is really the one who's moved this project forward in the past couple of years, and Francesca Fontana was the previous lab member who made the initial observations that I am going to tell you about, as well as collaborators. Now are funded by an NCI grant and receive support by local institutions, probably the Cyclone Cancer Center, St. Louis. So, to frame this presentation in a clinical context, just going to show you very simply the importance of bone lesions in essentially in breast cancer and in cancers in general. As you can see, the patients, the share of patients with skeletal-related events is very, very high. In fact, bone metastases are probably the most frequent complication of breast and prostate cancers. So clearly there is no question that the issue is important. It's also going only to grow in the future as more patients with those type of cancers survive longer. So our work has always been focused on cell adhesion within the bone microenvironment, and here giving a very simplified but important picture of the bone marrow neighborhood, essentially. And let me pick up my laser pointer here. So I wanted to draw your attention about the heterogeneity of cell populations populating the bone marrow. Of course, on the surface are bone cells, osteoclasts and osteoblasts, with their precursors, either hematopoietic stem cells or skeletal stem cells. And they interact widely through complicated paracrine loops that maintain homeostasis. Some of these interactions are mediated by adhesion molecules that allow cells to come in direct contact with each other and signal directly. But sometimes there are some uninvited guests in the bone marrow, tumor cells, which can colonize and sometimes hijack processes that are used in homeostasis for their own use. So there's a lot of interactions, as I mentioned. So here, this is a schematic view of digging these interactions a little bit deeper. So here on the right-hand side, you see a cancer cells. On the left, a stromal cell, and also on the lower left, an endothelial cells. As you can see, there are a lot of potentials for interaction through adhesion molecules. Shown here are cadherins, cadherin 2 and 11, which are the most important cadherins present in stromal cells and osteoblasts. Cadherin 2 is also called N-cadherin. This is the way we call it, and we'll continue to call it like that through the presentation. So depending on the type of cadherin that's expressed and on the differentiated state of these cells, different type of interactions can occur. And in fact, this is just a picture of cadherins and how they are part of the structure called adherence junction. The cadherin itself is a single transmembrane spanning molecules with a long 5 repeat extracellular domain and a shorter intracellular domain, which is very important because it interacts with several proteins of the cytoskeleton, some of which, however, are also important in signaling. For example, beta-catenin. We'll go over that in a rest of the presentation. Important is that the main cadherins in bone are cadherin 11 and N-cadherin. And N-cadherin has been attracted the interest of oncologists in the past because it is also important for the phenomenon called mesenchymal to epithelial transition, which is relevant to cancer progression. In general, the literature suggests that cadherin actually favors tumor progression because the overexpression in preclinical studies of N-cadherin in certain tumors, they become more aggressive, and inhibiting N-cadherin, in fact, inhibits growth of certain cancers. However, clinical trials using N-cadherin inhibitors have been relatively disappointing and not very uniformly conclusive. In fact, there is also evidence that cadherin can have an opposite effect. For example, in pancreatic tumors, removal of N-cadherin actually favors pancreatic tumor growth. And I wanted to draw your attention to the point that these preclinical studies were done using a genetic model whereby N-cadherin was deleted specifically in pancreatic cells. So this is a proof that N-cadherin can have different function, perhaps, depending on where it's expressed. So based on this biology, a group from Baylor College of Medicine years ago proposed this model, which is based on a nice study published years ago, whereby osteogenic cells in the bone microenvironment would essentially attract and function as a dock for cancer cells colonizing the bone through direct interaction between N-cadherin in the osteogenic cells and E-cadherin in the tumor cells. And they also trace this mechanism to activation of mTOR signaling in the cancer cells. So this is very nice and actually inspired us to get involved in this because we have been studying N-cadherin as important molecule in osteogenic precursors for many years and published more than one paper on this. And we had all the models to test this hypothesis in vivo. And so for these experiments, we initially did a very simple experiment, inoculating tumor cells in the mouse with or without N-cadherin specifically in bone cells. So to make these mice, we used the Osterix promoter driving Cre, Osterix being expressed primarily by osteolineage cells, although throughout the lineage, not just in differentiated cells. And then injected 2 tumors labeled differently, one in the mammary fat pad and one in the tibia, to test whether in fact the lack of N-cadherin would inhibit growth in the tibia, as suggested by the model that I just presented to you. But in fact, we could not reproduce or at least validate the model. So this is a bioluminescent scan of 2 mice with the 2 tumors injected. So the one in the tibia, as you can see here in the ovals, they seem to grow pretty much at the same rate. This is, I think, after 1 week of injection. And in fact, if you look down here on the X-rays on the right-hand side, you'll see the amount of destruction caused by those tumors is pretty much the same in the knockout and the wild type. If anything, there is even more osteolysis in the knockout. So no evidence that genetically removing N-cadherin from osteogenic cells, and I can tell you that this promoter is actually pretty good at deleting deeply in the osteogenic lineage. But there is no signs of even a reduction of disease burden. However, we noted that in the mammary fat pad, where we injected the other tumor just as a control, right? So this would be a normal microenvironment. Osterix is not deleted. N-cadherin is not deleted in the mammary fat pad. They seem to gradually grow bigger. And in fact, the tumor volume in those exoskeletal areas actually was significantly higher in the knockout animals. And also we started noting something unexpected, that there was a substantial signal in the thoracic area. So this represents clearly metastasis, but it was actually a signal much stronger in the knockout animals. And when we looked at the section, the lungs of these knockout animals were really full of metastases from the mammary inoculated tumors. You can clearly see the difference here. So that was actually a very surprising result. Not only we couldn't validate the idea that N-cadherin is necessary for homing of tumor cells to the bone marrow, but we saw perhaps the opposite. And these are data now published 1 year ago. This is 4 different experiments in addition to the one that I just show you in different conditions, and all are showing a significant difference in tumor size. So the question is, do they also, these animals lacking N-cadherin osteoexposed cells, do they also have worse metastatic disease? This is not conclusive, but it doesn't look like recurrence was, metastatic disease was really different in exoskeletal sites. However, the numbers of animals that we could follow is relatively modest. There was no difference in survival by Kaplan-Meier curve, but the subject risk was very low. However, median survival in the knockout mice was actually almost half as much as in the wild-type equivalent mice, suggesting that indeed tumor burden may be more severe in the absence of N-cadherin in these cells. Again, results were totally opposite of what we would have expected based on the model we just showed you. So the question, with this type of data is that how is it possible? How does N-cadherin in osteogenic cells control tumor growth at exoskeletal sites? You can understand that perhaps N-cadherin is dispensable locally, but why do we see an increased tumor burden outside of the skeleton? So there are potentially 2 explanations. One is the presence of factors that are produced by bone that act at distance. There are some data suggesting that, including this beautiful study showing that osteoblasts can actually mobilize neutrophils to enhance cancer growth in the lung. The other hypothesis, which is a little bit, out of the box, I would say, is that perhaps there are some cells that we are targeting in extraskeletal tumors, and therefore it is still possible that the red cell cell contact with these cells, if they exist in the tumor microenvironment, may account for the observation. So we were able to, to immediately test this hypothesis, the second one, with our mice, and in fact we used the AI9 cell tracker mouse to look at potentially cells outside of the skeleton that might be targeted. Here is that should show you a section of bone where Osterix is expressed. As you can see, it's present pretty much everywhere in all cells of bone, osteocytes, periosteal cells, osteoblasts on the surface, and in some cells of the bone marrow. So, be as it may, it is also present in breast cancers grown in mammary fat pad. So here I'm showing you an example of flow cytometry taken from tumors in control and Osterix targeted mice, showing that injecting either a breast cancer cell line or a melanoma cell line, we can actually see Osterix positive cells in the tumor. And some of them, probably a small proportion of them, also stain positive for N-cadherin. So this experiment that was supposed to be a negative experiment, essentially showing that no, there are no Osterix positive cells in the tumors, opened up a whole new direction of experimentation. So there are cells that express this would be osteogenic specific marker, and they also express N-cadherin. Work in collaboration with Roberto Civitelli in our center show that if you take these tumor-associated Osterix positive cells, isolate them, and then re-inject them in normal mice, along with breast cancer cells, the presence of these tumors-derived Osterix positive cells greatly enhances tumor growth. In a way, as it would be expected from what are called cancer-associated fibroblasts. Fibroblasts in the tumor microenvironment can facilitate tumor growth. So these Osterix positive cells do the same. But some of these cells are actually present also in soft tissues, typically sites of metastasis, without tumor. Yes, the percentages are very small. And here you can see again the flow cytometry picture of cells isolated from lung and mammary fat pad without tumor. There are Osterix positive cells, and you can see them also in these expanded cultures of cells isolated from the two tissues, only from mammary fat pad. So the number of these cells is very small. We now know that in the lung, these cells, there are actually some cells that look like type 2 pneumocytes that stain clearly positive, but there seems to be almost scattered throughout the lung, so with no clear pattern. So there are actually some resident cells that could potentially expand under stimulation of the tumor. Although we know for a fact that some of these cells in the tumor also originate from the bone marrow. So, so the question is, how does N-cadherin work in these cells to affect tumor growth? Is it because of the adhesion between cancer cells and tumor microenvironment cells, or is something else? Well, to to determine whether it's cell-cell adhesion, we used two approaches that will require adhesion. One is to look at gap junction intercellular communication. So gap junction communication allows diffusion of ions and small solutes and second messengers between cells directly through transcellular channels. And those transcellular channels can form only if two cells are physically in contact with each other. So we'll focus on this approach that we've taken, because I don't have time to show you everything. But just to give you an idea, one way of looking at the ability of cells to exchange, communicate directly through gap junction communication is to load cells with a fluorescent tracer that you clearly see, and then put the cells in contact with other cells that we can call acceptor cells, that we can see because of different fluorescent stain. So if gap junction communication is present, then the green cells will donate, will give green fluorescent dye to the red cells, and the red cells will become green or red. If there is no intercellular communication, we will see only either red or green cells. And the results are shown here when we put together, not necessarily co-cultured, but just mixed essentially, let these cells stay together for at least a couple of hours. Bone marrow stromal cells, green labeled, with tumor cells, red labeled. You can see that the red cells, the tumor cells, they're all green. They all show up in the green field. And the merged image show them to be yellow. So they have all taken dye from the bone marrow stromal cell. That's the only way they can become green. But the same as was this is wild type. In the cells taken from mice that don't have N-cadherin, you see the same. The red stained tumor cells are also green, which means that despite lack of N-cadherin, they can still adhere and form gap junctions. So the result, the conclusion is that N-cadherin does not prevent cell-cell adhesion. But that cannot be the mechanism by which N-cadherin enhances tumor growth. The other possibility is that N-cadherin interferes with signaling. That's not the canonical function of a cell adhesion molecule, but we know that cadherins can do that. And in fact, the literature, it's actually pretty rich about this. I'm just showing you one model that was published many years ago by some colleagues in France, showing that N-cadherin can interact with Wnt signaling components shown here. And in particular, it can actually bind to the LRP56 co-receptor, which binds Wnt agonists through an axing bridge, so to speak, and by doing this, it subtracts beta-catenin from signaling and channeling it into the degradation pathway. We've proven, actually we validated this model in our own lab and verified that in fact N-cadherin is a brake, restrains Wnt signaling in many different models. It also restrains even PTH signal transduction. So to understand whether the mice lacking N-cadherin in osteoexposed cells in the tumor might in fact have disrupted signaling, we used an RNA-seq approach and essentially isolate the cells from the tumor, remove the tumor cells first, and then remove CD45 positive cells, and use the non-hematopoietic population to isolate the osteorex positive from those that are negative, and compare the two populations using RNA-seq. This data, showing RNA-seq data usually not easy, but I just wanted to focus on this table here, and you can see that the differentially expressed genes can cluster around extracellular matrix metabolism and processing, essentially. The knockout cells seem to be more producing more collagen and genes that are important in extracellular matrix organization and degradation. Looking at specific markers, we find that these cells, as expected, compared to those that are osteolytic negative, do have an osteogenic phenotype. They do express genes, shown here in light green, that would be expected to be present only in osteogenic cells, particularly PTH receptor here, rank ligand, and collagen 24. So they do have genuine osteogenic footprinting, except alkaline phosphatase, which for some reason is actually downregulated. But that's not surprising because these cells, yes, can be osteogenic, but they don't make bone in the microenvironment. So they are non-mineralizing osteogenic cells. They express less of the markers that are used to isolate cancer-associated fibroblast. So they have some overlapping features, but clearly these are probably a different population in the stromal component than the classic cuffs. And the principal component analysis showed us that these positive cells, these positive cells actually cluster away from the negative cells, and that removal of N-cadherin pushed these cells more towards an osteolytic negative type of phenotype, suggesting in fact we're modifying the phenotype of these cells. And finally, to make again a long story short, we using different type of analysis of differentially expressed genes, we found that in general the knockout cells overexpress genes that are responsive to certain transcription factors, in particular SP1 and FAT and MES. They also overexpress LEF1, which is a direct component and actually the target gene of canonical Wnt signaling. So this finding here validates and confirms that N-cadherin is an inhibitor of Wnt signaling, even in these cells in the tumor microenvironment. But clearly there is far more in terms of signaling modulation by N-cadherin than would be anticipated by simply interfering with Wnt signaling. So in the next phase of the experimentation, we focused on these mechanisms. So to summarize the results of these studies, we find that N-cadherin in these particular tumor-associated osteolytic cells, cells that we have identified that were not known before, restrains tumor growth at extracellular sites. It does not alter the tumor microenvironment cell adhesion, but it downregulates a pro-tumorigenic transcriptional signature in these microenvironment cells. I should tell you that this function here, again, is at variance with what's known about N-cadherin function in cancer-associated fibroblasts, where it is believed to actually do the opposite, favor tumor growth by acting and activating cell migration. So clearly, depending on the cell context in which the protein is expressed, the function can be radically different. So the next question we wanted to ask is which of the signaling pathway I just shown you, which could be affected by N-cadherin, are the ones important for this anti-tumorigenic activity. So this mechanistic study, we had to rethink our approach because, unfortunately, as I've shown you from the FACS analysis, these cells are rare, and in fact, we tried to culture them and failed. Apparently, it may be possible. Roberto Civitelli in our center with her group has been able to culture these cells by collecting a lot of tumors and expanding them first, but they are not very easy to work with. So we turn to an embryonic mouse cell line, the MCT3E1, which I think is a good surrogate for these particular studies. Of course, we'll have to validate back into the real cells, but these cells are very similar to the osteoresponsive cells in the tumor because they have an osteolineage phenotype, although they are not differentiated. They express both osteolysis and N-cadherin and all the phenotypic features of osteogenic cells, and they can be induced to differentiate into osteoblasts. And most important, they respond to factors involved in the tumor bone cell so-called vicious cycle, and in particular, Wnt agonists and antagonists, but also members of the BMP TGF-β superfamily, as well as BTHRP. And finally, again, as I mentioned to you, important in this particular approach is that N-cadherin is known to restrain Wnt and PTH receptor signaling in osteogenic cells. So this is again, a simplified picture of what happens in the bone microenvironment. That's where we started, and now we are thinking about tumor osteogenic cells interaction in extraskeletal tissue in the breast. But the type of interactions between these two cells, between the resident cells and the tumor cells, are probably very similar in the bone proper as well as in extraskeletal tissues. And I think the players are probably the same. We know that the tumor cells respond to factors produced by osteoblasts, and in particular TGF-β, which is a good stimulator of tumor cells. The tumor cells in themselves produce factors that can stimulate or inhibit osteogenic cells. So the result of all this is typically a situation where tumor cells can grow very well. So for this experiment, we actually focus on TGF-β because, as you can see here, it's really central and can be produced by osteoblasts. It can be also released by osteoclasts from TGF-β that's embedded into the matrix. And it's a very good tumor stimulator. So based on that biology, so first of all, we created a cell lines using the MCT3, lacking N-cadherin, using the CRISPR-Cas9 technology, which makes it very easy to create knockout cell lines, and generated several cell lines with N-cadherin knockout. And then we used these cells for two things. First of all, to look at signaling using molecular biology tools, and also look at their effect on growth of breast cancer cells. Sorry, using 3D co-cultures in vitro. So just to validate the system, when we co-culture these cell lines, whether wild type or lacking N-cadherin. By the way, you may have noted that the N-cadherin gene is called the CDH2. Okay, sorry, I forgot to mention this quirk in the nomenclature. So cadherin 2 or CDH2 is N-cadherin. So as you can see, pretty obviously is that the tumor cells, which are visible because they are GFP tagged, in the presence of the osteogenic cells, can form these spheroids in co-culture, and pretty much all of them have tumor cells, essentially. So these are primarily tumors. But they clearly grow much faster in the presence of N-cadherin knockout osteogenic cells. To quantify this, I'm showing you here the fluorescent intensity as a parameter of breast cancer cell growth. These cells grow very well, even in a small concentration of serum on their own. But if co-cultured along with wild-type osteoblasts, the growth is more than twofold higher, and in the presence of osteogenic cells lacking N-cadherin, the growth is even higher. This is very highly significant because the scale is essentially it's pretty large. So this is with the 41 tumor cell line. The same result, almost identical, with the more aggressive B01 cell line. And, larger growth alone, enhanced by the presence of bone cells, further enhanced by the presence of N-cadherin deficient bone cells. So, what are the signaling pathways that can cause this? Certainly, Wnt signaling can be one, but the proteomic analysis also pointed us towards the PI3 kinase AKT pathway specifically. So we hypothesized that this pathway can be in fact a key element in N-cadherin regulation because it can activate both SP1 dependent genes and Wnt signaling dependent genes by non-canonical stabilization of beta-catenin. So an increase in LF1 and SP1 would match exactly what we have found in the proteomic analysis. So we set out to look at to test this hypothesis that N-cadherin may in fact interact with components of the protein, the PI3 kinase, by essentially borrowing the idea from what happens with Wnt signaling. As I was showing you earlier in the presentation, N-cadherin can bind beta-catenin and in that way inhibit Wnt signaling. So we asked whether it can also bind key components of PI3 kinase, thereby sequestering them out from signaling. So first of all, we using these cell lines we just created, we were able to in fact confirm that specific signaling pathways are in fact hyperactive in the knockout cells. And we use TGF-β as a stimulator because of it's important in the tumor vicious cycle, as just explained to you. So these are it's a complicated compound Western blot. I just want to point to essentially the essence. So you have here control and N-cad negative cells, and you can see here that the cell line is really knockout as far as N-cad, and that we have a pretty uniform loading. So upon stimulation with TGF-β1, there is a fast activation of PI3 kinase, here shown as phosphorylation of P85, and this phosphorylation is far stronger in the knockout cells. Okay, no effect on total P85. Phospho-AKT is also stronger, more strongly activated by TGF-β in the knockout compared to control, with no effect on total. And also phosphorylation of beta-catenin in serine 552, which again stabilizes the protein, as opposed to the phosphorylation sites that are dependent on GSK3. And here again, probably a faster activation of beta-catenin. So overall, lack of N-cadherin does enhance not just Wnt signaling, but also PI3 kinase signaling. So Toshi, who's done most of this work now, also used the co-immunoprecipitation to test the interaction between N-cadherin and the P85 as a key component of PI3 kinase. So as you can see here, there is evidence of interaction between P85 and N-cad. using N-cadherin antibodies for immunoprecipitation and the P85 antibody for Western blot, and doing the reversal, you see the same result. N-cadherin can also bind to the other component, P110. So no question that N-cadherin can bind PI3 kinase components, and by that it can also upregulate. Actually, lack of N-cadherin can lead to upregulation of downstream signaling. Okay, and this is demonstrated here. So here we're looking at mRNA expression of SP1 and LEF1. As you remember, those are the downstream result of activation of PI3 kinase, and this is the relative expression compared to wild type in the knockout. And in both cases, there is a 2 or even 5-fold increase in expression of these two genes, suggesting again that by removing that interference, that break on the PI3 kinase, the signaling system is hyperactivated. And finally, we actually look at production of TGF-β1. Okay, now here we use TGF-β both for activating the cells and also as a readout of a factor that we know it's important in tumor growth. So TGF-β in these cells clearly functions as an autocrine mechanism. It can stimulate its own expression. It does so in wild-type cells, but more so in cadherin knockout cells. And this is all significant, suggesting that, in fact, lack of N-cadherin is a significant enhancer of TGF-β1 dependent TGF-β1 production. And how does this play out in terms of cancer growth? Can TGF-β1 in fact be one of the main mechanisms by which these microenvironment as osteogenic cells stimulate tumor cell growth? Okay, this part of this cycle was not known, although we know that tumor cells can respond to TGF-β very well. So to test whether TGF-β is in fact important in the proto-tumorigenic effect of osteogenic cells, we created another set of cell lines using the same CRISPR-Cas9 system to generate breast cancer cell lines lacking the TGF-β receptor 1, which is one of the most important component of the heterodimers that transduces the signal from BMP and TGF-β. And TGF-β receptor 1 seems to be specific for TGF-β more than for activins or BMPs. So we created this TGF-β, let's say, resistant cell lines, tumor cell lines, and generated again the 3D co-cultures with wild-type osteogenic cells. And the results are pretty striking and pretty clear-cut. Not the usual result in our lab, where most experiments don't work, but this one worked pretty well. So here I'm showing again two different breast cancer cell lines, the 41 and the BO1. The first box plot here is fluorescence intensity, which is essentially tumor growth in the presence of 5% FPS of tumor cells. This is the growth of breast cancer cells alone, but without the TGF-β1 receptor. You already can see that we are suppressing growth substantially, implying that perhaps, in fact, TGF-β could be even an autocrine growth factor produced by tumor cells themselves, which is an important consideration to make. Now, if we grow these tumor cells in the presence of the osteogenic cells, as we've seen before, there is a twofold increase in growth. But if we co-culture the knockout cells, lacking TGF-β receptor 1 with osteoblasts, this enhancing effect is completely abolished. And in fact, growth is even lower than in the absence of osteoblasts, suggesting that TGF-β is really a key factor driving growth in the cells, and that's probably a key element in mediating the proto-tumorigenic effect of osteolineage cells. And the results with the BO1 cells are exactly the same. So, how do we put all this together? And this is my concluding slide. There should be some time for questions and discussion. So, essentially, what we have discovered is, rather feed-forward or positive feedback loop based on TGF-β. So here is schematized, how that may work. So this is tumor-associated osteoexposed cells. So this is tumor cells. This is N-cadherin in its conformation of a cell adhesion molecule. So we know that TGF-β signaling activates the PI3 kinase and AKT. So this is what happens when we challenge these cells with TGF-β, activation of AKT, activation of both SP1, and stabilization of beta-catenin. This results in downstream activation of genes that also lead to secretion of TGF-β1. Okay, so TGF-β1 is again the product of this genetic regulatory system. TGF-β1 goes into the tumor microenvironment where it activates cell growth by binding to the receptor in the tumor cells. And from here, the situation becomes probably more complex than I depicted here, but it's likely that TGF-β itself may be one factor that feeds back into the tumor-associated osteoexposed cells, although clearly there are other factors that are secreted by tumor cells that can activate PI3 kinase or other signaling system like Wnt signaling that are inhibited by N-cadherin. So a larger tumor mass upon activation by TGF-β releases more of these factors which feed back on microenvironment cells, which again continues to produce tumor activating factors. So that's how these tumor cells, this microenvironment cell can control tumor growth. And N-cadherin works as a brake to this system. In this case, shown here by sequestering out key component of the kinase system activated by TGF-β. But again, there are certainly other signaling systems that are affected. So, overall, this study shows that these osteogenic cells, or cells that have an osteogenic signature present even in extraskeletal tumors, represents a system very similar to what happens in the bone marrow microenvironment in the presence of cancer metastasis, except that this system is active in the primary tumor as well. And we don't know why these osteogenic cells should be there and how they are recruited to the tumors, or whether they result from expansion of local cells. But clearly, these are a different type of microenvironment cells, different from the CAF, that have specific functions in tumor growth. And of course, if we learn more about this, we could find now different potential targets for interfering with this proto-tumorigenic action. Perhaps we can target N-cadherin more selectively in tumor itself, or perhaps we can develop more targeted anti-TGF-β1 therapies. So This was my last slide, and thank you for your attention, and I'll be glad to answer any questions.
Thank you very much. If you have any questions, you can either or you can type it in the chat box and we can read it and we'll answer. One of the questions that I had was, do different tumor types of tumors have different types of osteogenic cells, or the expression of cadherin in them, are they different?
I do not know. We haven't really checked on that. In fact, one of the current experiments in the lab is to, in fact, look at human tumors. We are using a tissue bank repository that our Sightman Cancer Center has banked with different tumors, and we're exploring application of spatial transcriptomics approaches to essentially answer the question: are these cells present in all tumors, first of all, and if there are they different depending on the tumors, and is there a correlation between N-cadherin expression in these cells and tumor grading, essentially, severity of the disease? So I cannot answer the question directly, but it's an important one. There's somebody. Yeah, Girish asked a question in the chat here. Thank you for the presentation. I'm wondering how you plan to envision translating your research to human bone cancer therapy. Yeah, of course. Yeah, as I just mentioned, I think if we learn, first of all, the presence of these cells opens up a whole new host of potential targets, right? The question is how to, for therapeutic purposes, how do we target selectively these cells in the tumor microenvironment? So that's going to be the challenge. Perhaps using nanoparticles, we may be better shaped to drive potential, inhibitors to bind to N-cadherin in these cells. And the other potential application is, if not in therapy, would be in diagnostics. Can we rapidly develop a diagnostic test based on simple histology to predict the clinical development of these tumors using the markers I just shown you? So those are the potential translational directions. Keep in mind, acknowledging that is going to take a while before we get there, and that we still need to work on understanding how these cells really work and how they get there, if in fact they are recruited. So another question is, let's see, pancreatic cancer, especially early diagnosis, would be great. Yes, I agree. Yeah. So again, developing potential tools for diagnosis is one exciting development that we're hoping to develop. Right now, we'll be focusing primarily on breast cancer with the human translation to make sure that we understand how it works there. Then can probably expand to other tumors. Another question by Stephen Winters is, does cadherin expression in bone vary? Are there effects of sex steroids, HGH, vitamin D, etc.? Very good question. Yeah, so that's been asked the question very frequently. So we know that N-cadherin expression varies during the osteogenic lineage differentiation. It's expressed at a higher level earlier on in cells that are not yet differentiated, and that it is downregulated as osteoblasts become more and more matrix-secreting cells. And it is actually replaced by another cadherin, cadherin 11. However, N-cadherin is very important in keeping osteoprogenitors in their osteoprogenitor state, because if we remove it at that stage, we have demonstrated that the number of osteoprogenitor cells decreases substantially. So this is the way N-cadherin is regulated in the osteogenic lineage. Are there effects, effect of hormones that can, therefore, can vary in certain pathologic conditions? We do not know. We haven't really done formal studies on even sex differences, so I cannot answer the question.
Has this cadherins anything to do with atherosclerotic plaques and the calcification there? And is there anything that these might play a role in those?
Atherosclerotic plaques, yes. Well, yeah, so long as, in certain conditions where calcification are in fact the result of activation of the osteogenic differentiation. I know that happens, for example, in calcification from chronic kidney disease, probably less in atheromas themselves, at least initially. Yes, I would envision potentially a role in that case, yes. So that could be a good way of, could be a potential, direction to at least explore the possibility preclinically. Something that I thought about doing many years ago, but then drifted away from it.
And is it also, are the osteogenic cells normally present in a wide range of tissues?
That's another very, very good question. So, I am, just one of the many investigators in the field to ask it. As we developed and used these Cree drivers, hoping that they would be very cell type specific, we are now finding that it's the expression is probably broader than we had anticipated, and that applies pretty much across the board. In the particular case of Osterix, it's been particularly challenging because we thought it would be a very, very specific for bone and cartilage, but in fact, it's probably not exactly the case. There have been earlier reports from other labs suggesting that there are Osterix body cells, for example, in the intestine, and I think in the ear, and not in bone cells of the ear. We are finding, as I've shown you, some cells in the lungs, in the mammary fat pad. So, I mean, where else would they be? It's possible that there are also in other tissues. So, but I can't say that as yet. So, yeah, and the question is that I've been asking, what do they do there?
Any other questions, comments, anything? Well, I would also like to mention that Dr. Civitelli will be presenting medical grand rounds tomorrow, talking on the treatment of osteoporosis, sequential and combined therapies. So we invite everyone to join that also tomorrow from 8 a.m. to 9 a.m. for medical brand rounds. Thank you, Dr. Civitelli.
You're welcome. Thank you for listening to me.
Yeah, thank you very much. Excellent talk.